Hi, this is our second lecture on proteomics, where we'll go into more detail building on the information that we provided in the first proteomics lecture. First of all, we briefly introduced various amino acids last time, so for those of you who are not familiar with the 3-letter or 1-letter code for the 20 common amino acids that constitute proteins in most biological systems, they're listed here for you. You'll notice that many of the we typically use single letter code, and single letter code is usually the first letter of the amino acid name. Although since there are multiple ones with multiple amino acids that start with A, there are other letters that need to be used. Last time we talked about how we acquire MS and MSMS data on peptides for protein identification and proteomics analyses. We'll want to expand upon some of that information. One thing to keep in mind is that in all MS analyses, whether we're analyzing data through automated searches or through manual evaluation, we are comparing observed and expected masses. Observed must equal expected mass within plus or minus the error of the measurement. In most current measurements, 1 Dalton or less is very critical, except for very low mass accuracy analyses. This means we need to know whether we're dealing with mass M or protonated mass MH+. There are also other cases where we might be talking about doubly charged mass of a peptide or protein. That would be M H2 plus. Also, are we talking about monoisotopic or average masses, both for the calculated and the observed values? In addition, factors such as whether cysteines are present in the protonated form or as disulfide bonds are critical because the mass difference is approximately 1 Dalton. Cysteines are invariably modified in most proteomic analyses because cysteines are sensitive to oxidation, and we can modify them. Two common reagents that are used are iodoacetamide or iodoacetic acid. These also differ by Dalton. If we don't have the correct form of these various masses or know whether we're talking about M or MH+, we can obtain an incorrect interpretation. The difference between monoisotopic and average masses depends upon the size of the peptide or protein, as illustrated by this table, which indicates the difference between the two. Mass errors are typically reported as parts per million and can be calculated as indicated here. As illustrated, if we're in the low parts per million range, as is often the case for modern ESI-MS measurements, this means the third and fourth decimal for a peptide of approximately 2,000 Daltons is significant, and therefore we will typically work with high mass accuracy data where at least 4 decimals are reported for peptides. An advantage of high mass accuracy data is amino acids and amino acid combinations that might be isobaric at low mass accuracy are rarely isobaric at high mass accuracy. An example is lysine and glutamine, K and Q, which differ by 0.03638 Daltons. If I have a low mass accuracy measurement, I cannot tell the difference between these. However, if I have a high mass accuracy measurement, the difference between these becomes quite obvious, as indicated by these mass errors indicated above. Nonetheless, there are some situations where I have an exactly isobaric situation. One is the amino acids leucine and isoleucine, which have exactly the same amino acid composition and therefore are identical. The only way you can distinguish between leucine and isoleucine is with a high energy fragmentation that would actually fragment the side chains. Other considerations with high mass accuracy data are that subatomic particles do not necessarily sum to the masses of the major component parts, such as neutrons, protons, and electrons. And therefore, we need to use exact masses, as indicated here. A monoisotopic shift is actually a neutron, but the neutron in C13, the monoisotopic mass difference is slightly different than that of a neutron. On the other hand, when adding or calculating masses for organic molecules, molecular masses are always the sum of the component atoms, and therefore if I know the masses of the individual atoms, I can calculate amino acid masses or mass changes resulting from deamination and so forth, as indicated here. We briefly mentioned that proteolysis is frequently performed using trypsin. One question is, why is it a popular choice for LC-MS/MS analysis? One reason is that the average peptide size is ideal for both reverse phase HPLC separations as well as for ESI mass spectrometry. That is, the average peptide is neither too small or too large to be effectively separated and analyzed by an LC-MS/MS experiment. Most tryptic peptides are also have a +2 charge in electrospray experiments, and this yields a nearly complete singly charged series of complementary B and Y ions, which facilitate peptide identification. Trypsin also exhibits a robust proteolysis. It is not particularly sensitive to protein conformation or adjacent sequences, and it's highly specific for cleaving the C-terminal side of lysine and arginine. It also cleaves nearly all lysine and arginine bonds. There are a few cases where cleavage is incomplete, lysine and arginine followed by proline, or where we have clusters of lysine and arginine in a contiguous series. It is also relatively cheap and a highly purified protease. One question would be, if I have a monoisotopic mass of a protein which doesn't contain cysteine, so I don't need to worry about whether cysteines are in disulfide bonds or not, and I have the indicated mass. A mass analysis of the intact protein yields a value of 25,711.88. What additional information would you need to determine whether this mass is consistent with the protein sequence without modifications? The correct answer is B, I need to know the mass error. Otherwise, I don't know if this is within the mass error. I also need to know whether I'm talking about M or M plus H. In such a large protein, 1 Dalton may not be that important, but if I'm near the edge of the mass error, it could be important. And therefore, this also is, so I need this, and I need this, and I also need to know if it's an average or a monoisotopic mass, and therefore the correct answer is E. The three major types of protease studies, key factors in selecting an appropriate experimental approach are summarized here. Protein profile comparisons are quantitative comparisons of 2 or more proteomes or subproteomes. Key question is, what's the depth of analysis needed? Another is, what's the best way to quantitate? And what is the associated with this? What's the minimum fold change desired? For protein composition, I want to identify as many proteins as possible in the proteome or subproteome. I therefore need to know the depth of analysis. I don't need quantitation here. The biggest issue is going to be distinguishing contaminants from low abundance components. Protein-protein interactions are a third type of proteomic study, which we won't talk about in much detail here due to lack of time. However, I can do a binary protein-protein interaction analysis, or I can look at large protein complexes. The major challenge here is distinguishing contaminants from low abundance, low affinity components. So selecting the best proteome analysis strategy for protein profiling or compositional analysis, keeping in mind that the only difference between these two is for compositional analysis, I don't need to worry about quantitative issues because I'm not comparing two or more samples. Key question is, what is the goal of the experiment? What's the best approach? Am I going to look at targeted or global proteomics? And do I care about identification of post-translational modifications or chemical modifications? For most post-translational modifications or chemical modifications, I need to use an enrichment strategy to selectively purify either the proteins or the peptides that contain these desired modifications. What is desired depth of analysis? Usually I can increase the depth of analysis by performing more fractionation, but this decreases throughput and increased costs, and therefore we may need to make some compromises and settle for a less than complete analysis of the proteome so that we can maximize throughput and manage time and costs. Another consideration is what is a minimum fold change that I am interested in? Do I need to have 1.5 fold precision, or am I only looking for very large changes? The types of methods that we can use for protein profiling or compositional analysis are summarized here. Top-down and middle-down applications are defined, but are relatively specialized and are not as commonly used as the analyses where proteins are converted to tryptic peptides and analyzed after fractionation by one of several alternative methods. There is a common method called bottom-up analysis where the entire protein mixture is digested with a protease separated and analyzed, usually because of the complexity to achieve a good depth of analysis. We need to have multiple fractionation steps. So LC-LC-MS is two types of chromatography, or IEF followed by LC is isoelectric focusing separation of peptides followed by LC MSMS. These bottom-up approaches are quite viable. However, they are limited to some extent by the fact that most tryptic peptides have a limited range of physical properties that one can exploit in the two-dimensional peptide separation. Hence, combining protein and peptide fractionation is the most powerful approach for achieving maximum depth of analysis. Basically, we separate proteins by using one or more fractionation methods. We then trypsinize these individual fractions and then analyze the peptides by LC-MS or less commonly LC-LC-MS. A powerful, simple strategy is oftentimes referred to as gel-C-MS, and this is separation of the proteome by one-dimensional STS gels, which are easy and run in most laboratories. We then cut the entire gel lane into uniform slices, digest each slice, and conduct LC-MS. One thing you'll note in all of these procedures, the last step in all of them are LC-MS. So the variation is, do I first digest the protein into peptides, or do I separate proteins first? And what method of separation, if any, do I have before the LC-MS step? Protein compositional analyses, the goal is to identify as many proteins as possible in the proteome or subproteome. We can use any of the protein profiling methods that were listed previously that utilize LC-MS as a final step. The only difference is quantitation is not needed. Two-dimensional gels and 2D dig, which we'll briefly discuss later on, are not appropriate techniques for compositional analysis, and this is because 2D gels are primarily comparative methods where I compare the two-dimensional patterns of proteins from two or more samples. I then look for differences. I do not have an identification inherently as part of the analysis. And if I separate a proteome on a standard format two-dimensional gel, I might generate 1,500 to 1,000 spots. Cutting out 1,500 to 2,000 spots. Cutting out 1,500 to 2,000 spots is, and digesting them and analyzing them is an extremely low throughput approach. And therefore, I would never do a compositional analysis with 2D gels. So our second question, mitochondria are isolated from a human cell line grown in media containing fetal calf serum for proteome compositional analysis after trypsin digestion. What contaminants could be difficult to distinguish from low abundance mitochondrial components? So I've isolated mitochondria. The ability to analyze this proteome and distinguish it from contaminants is going to be dependent upon my ability to identify proteins that originate from mitochondria from proteins from other sources. Distinguishing the mitochondrial proteins from cytoplasmic and nuclear proteins would be very difficult. So this is a good answer. What about fetal calf serum? Fetal calf serum, while some peptides may be identical to human serum proteins or other human, human proteins in the database, there are going to be sequence differences, so I can really distinguish these. So going back, this was a good answer. This was a good answer. But fetal calf serum proteins I can distinguish based upon sequence differences between bovine and human. Trypsin is from a non-human source, and so I can distinguish that. So the best answer here is A. Common bottom-up proteome analysis strategies have the common feature that we initially digest the sample with trypsin or another protease prior to any fractionation. All these methods are compatible with SILAC or chemical labeling. We'll explain more about these labeling methods in a few moments. All methods use LC-MS as the final step. The difference, therefore, is what's the first step. The most common methods use strong cation exchange or isoelectric focusing. Isoelectric focusing is most effectively done using an instrument called off-gel electrophoresis. And the third option, which is relatively new, is using high pH reverse phase separation. All of these are the first step. The second step of separation is the low pH reverse phase part of the LC-MS method. Briefly, the LC-LC method using strong cation exchange is one of the oldest techniques, bottom-up techniques, and it is sometimes referred to as MUDPIT for multidimensional protein identification. It utilizes strong cation exchange of the tryptic peptides, and this can be done offline or to achieve more sensitivity can also be done in an online mode as diagrammed here. The alternative to SAC's LC-MS is ion exchange. The off-gel electrophoresis approach uses a solution-filled multi-compartment plastic grid, which is placed on top of an IPG strip. The IPG strips are the same types of strips that are used as in the first dimension of two-dimensional gels, except here we're separating peptides instead of proteins. And this is a pretty robust method. Another alternative is to use high pH reverse phase followed by low pH reverse phase, and this is superior to off-gel in terms of resolution, and also off-gel electrophoresis is the most expensive of the bottom-up approaches. Somewhat surprisingly, the use of both high pH and low pH for reverse phase actually achieves very good separation, despite the fact both are reverse phase separations. And this is because at different pHs, peptides have different charges, which affects their hydrophobicity. This is a relatively new technique, has not been extensively used so far, but probably will expand in utility as more laboratories migrate to this technique. In terms of combined protein peptide fractionation, gel LC-MS has been shown to be the most efficient technique, as indicated by two 2010 publications, one of which is referenced here, and the data from that publication are summarized here. As you can see, by multiple criteria, using SDS separation is better than solution IEF or IEF, IPG-based IEF, or the strong cation exchange approach. Some of the advantages of gel LC-MS are it's simple, universally used sample preparation method that's very compatible with very low protein amounts. We don't have to worry about losing the protein because we're in SDS, which minimizes or avoids, essentially avoids the risk of absorptive losses. We have the greatest depth of analysis, as indicated by the study cited previously. Protein size information is also preserved. It is also easy to expand the depth of analysis by simply using more gel slices. And the disadvantage is that it is not particularly compatible with peptide isotope labeling methods due to slight variations in separation from one gel slice to another. A couple of examples from our laboratory are summarized here. These are human NK cells where 500,000 cells have been 500,000 cells have been separated on each of the gel lanes, and we cut 30 slices as indicated, combine the results from corresponding slices from two lanes, and digest and analyze, and with a low false discovery rate can readily identify 3,000 to 4,000 proteins. Similarly, we can look at secretomes from samples such as ovarian tumors and obtain thousands of proteins from a moderate number of gel slices. So question 3 is mud-pit analysis of human cell line identifies ubiquitin, which is an 8K protein, as one of 2,000 proteins. Another analysis of the same human cell line uses gel LC-MS and identifies ubiquitin at 8K, 125K, and 220K. And it identifies a total of 2,800 proteins. In terms of ubiquitin-related biology, what factors make gel LC-MS the best choice? More total proteins are identified. While this is true, it doesn't give us more insight into the ubiquitin-related biology. Running 1D gels is easier than ion exchange, perhaps, but again, it doesn't give us insights into ubiquitin biology. But question C, or answer C, ubiquitin is present in the unconjugated 8K form and also in two specific protein bands. This is an important consideration because now we have information about two proteins that are modified, whereas the peptide analysis does not give us, the bottom-up mud-pit analysis does not give us this information. And therefore, the best answer is C. Now, to go into greater depth in terms of the quantitation methods, if we can divide these into three different categories. quantitation where the label is introduced pre-trypsin digestion, which is SILAC. SILAC stands for stable isotope labeling at cell culture. We're metabolically labeling one of multiple samples to be compared with heavy isotopic amino acids and using normal isotope labeling for the other sample. It's most commonly done in a 2-plex, but a 3-plex is possible. Post-trypsin labeling is not an optimal method that involves protein fractionation because it is insensitive to any variations that occur at the protein fractionation level. This involves chemical modification with stable isotope tags. The most common are multiplex reporter ions such as iTrack or tandem mass tag. These tags are isobaric in the MS spectra and can be multiplexed at various levels depending upon the specific reagent set that's used. The third and simplest method is label-free analysis. The simplest of these is to use spectral counts, which is simply how many spectra match a given protein, and the other is integrating MS ion currents. SILAC labeling was first described by the Matthias-Mann group. Typically, lysine and arginine are labeled. 2-plexes use normal abundance, lysine and arginine indicated by K0 and R0, as well as lysine and arginine in the second sample, which is 6 Daltons heavier due to the presence of C13 atoms in these amino acids. The 3-plex is also possible using the isotopic abundance, using the stable isotope amino acids as indicated by the increased masses. The analysis is basically like any other LC-MS/MS experiment, except that we look for peptide pairs that differ by the expected mass but coelute. This is an important consideration. Stable isotope labeled peptides are chemically identical to the normal abundance peptides and therefore coelute on the reverse phase separation. We therefore are looking for mass differences at the same position in the HPLC gradient. We calculate ratios of the precursor ions, heavy over light, and average the ratios for all measurements matching a protein. The pros and cons include the fact that it's ideal for it's ideal because the 2 samples are mixed before any processing step, so it is insensitive to any variations that occur throughout the processing of the sample. It is, however, largely limited to experiments where saturation metabolic labeling is possible. These are usually cell cultures or growing microorganisms. However, some alternatives have been described. One is that you can label entire organisms by feeding them heavy amino acids, and this has been done with several mouse models. Another is a super SILAC approach, where one takes a series of cell lines that represent a non-labelable source and mix them together and use them as a reference. The iTrack and TMT are amine reactive isobaric labeling tags that can be multiplexed. We react each sample with a specific reagent after trypsin digestion, and then mix 2 or more labeled tryptic digests. A single peptide is identified in MS scan from all samples because all tags have the same mass. The tags then fragment in the MS step, and different reporter ions are seen as indicated here. So here I have 6 samples that have been labeled with the 6 different tags, and at this stage, and I now have different reporter ions as indicated, 120, ranging from 126 to 131 Daltons. So we can do extensive multiplexing, which saves instrument time. However, this is primarily limited to methods that do not involve protein fractionation, since any variations in the protein fractionation will be detected as changes between samples. This is also true at the protease digestion step. There are several label-free quantitative methods that also can be used. Sequence coverage has historically been used occasionally, but it is a poor method, and hence the reason why it's grayed out. The linear range of this method is too narrow to be very useful. Spectral counts is based upon the premise that as the amount of a protein increases, more MSMS spectra will be performed and matched to peptide sequences for that protein. This is a wider linear range. It's very quick and easy to quantitatively compare. However, it is less accurate than most other quantitative methods. Also, it is not very effective at very low concentrations. The last label-free method is to integrate high mass accuracy, high resolution MSMS ion signals. It's the most accurate of the label-free approaches, but it is much more complicated to perform compared with the spectral analyses. It requires dedicated and complex software. One of these is Rosetta Elucidator software, and this shows a comparison of technical duplicates. The data that's actually being compared are these two-dimensional signals as shown here. The x-axis is retention time, and so this is the reverse phase HPLC separation, and at each specific point in the chromatogram, the masses over charges are plotted ranging from over the range that has been acquired. The signals are all the ions that are detected. The intensity of the signals are color-coded as indicated at the top. We can use this type of an approach to compare multiple samples such as serum. In this study, serum from patients with ectopic pregnancy versus patients with normal pregnancy were compared, and these are the quantities for multiple peptides matching a given protein, and you'll see that in all cases the peptides are signals are higher in the ectopic pregnancy versus the control. These are the MS2 peaks that are integrated to give the values in the top panel. So spectral counts are simple but the least accurate, and they are particularly inaccurate for low abundance proteins because low abundance proteins will have very few peptides identified and very few spectral counts, and a lot of variation between replicate analyses. An advantage is that there's no expensive tagging reagents used, and we do not have to worry about completeness of the tagging chemistry. They cannot be directly multiplexed, therefore the total mass spectrometer time is increased. For example, if I have 20 fractions times 6 proteomes, and I run an isobaric tag experiment where I have a 6-plex, this could all be done in one experiment for a total of 20 fractions, whereas in this case, with a label-free approach, I'll have 20 fractions times 6 proteomes or 120 MSMS runs. This method is sensitive to any experimental variation, but internal references can partially control for this. Minimum fold change is usually larger. Minimum fold change that can be considered to be significant. A significant change is much larger than for tagged methods. So you have a research project that involves quantitative comparisons of proteomes of a strain of E. coli grown under 4 different culture conditions and using gel LC-MS. Which quantitative method would be the most appropriate? So we could use SILAC because it's E. coli, so we can metabolically label. So this would be a possibility, and it is early, so we don't have to worry about any variations during the protein separation. iTrack or TMT are a poor choice because variations in the gel separation complicate the analysis. Spectral counts could also be used. but will be less accurate than SILAC. So it's not as good a choice as SILAC. Label-free integration of MS ion signals, again, could be used, but will be less accurate than the SILAC. So all are equally appropriate. While one could argue that the label-free methods could be used and have compensating advantages over SILAC, iTrack is not compatible, so E is not a good choice. Therefore, the best answer is A. There are several additional protein profiling methods that warrant a brief mention. The one is two-dimensional gels. This is the oldest proteomic technique, and it was in use before proteomics was even developed as a field, as mentioned in the previous lecture. It separates proteins by isoelectric focusing on an immobilized pH gradient, followed by an SDS slab gel. It has major limitations. It has largely been replaced by 2D DIG, which is the same as 2D gels, but here 2 or 3 samples are labeled with psi dyes, where the size of the dye and the charge has been balanced, so that migrations are similar on the 2 dimensions. It's better than the standard 2D gels because we don't have to worry about gel-to-gel variations, but other limitations of 2D gels persist. These other limitations include difficulty in identifying small proteins, large proteins, very acidic, very basic proteins, as well as hydrophobic proteins. Another method, which we won't describe in any further detail, is antibody arrays. This is a sensitive, high throughput method, but it's not discovery based, and it really is limited by the availability of antibodies that give linear responses in parallel analyses. A little further detail on two-dimensional gels. Pros are it can detect changes in charge and size, and therefore post-translational modifications. Isoforms, such as alternative splice forms, can potentially be detected, but it doesn't identify all proteins. You're simply looking at spot changes, and it is another advantage is you can archive the images and compare many different samples over a long time frame. A disadvantage include the gel-to-gel variation, which is largely eliminated by the DIG approach, but the other disadvantages persist. I've already mentioned the poor representation of certain groups of proteins. It is also very tedious and requires a high skill level to perform the analyses with high quality. Most important limitation, perhaps, is that you only see the most abundant spots, and you tend to see on the order of 1,000 to 1,500 spots, perhaps as many as 2,000 on the best 2D gels. However, this is spot number, it's not gene products. The number of gene products can be quite a bit lower because you are seeing multiple forms of some proteins. This is a brief summary of the overall 2D gel analysis. This is a representative two-dimensional gel, and if you zoom in a specific region, you'll see that you actually have a large number of spots that are identified. Frequently, spots in a train, such as this one, are different charge states of the same protein. One cannot assume that this is always the case, but one can cut out the spots and identify them using mass spec to verify whether or not this is true. 2D gels are very sensitive to dynamic range, and so protein samples which have a wide range of protein abundances, such as human plasma or serum, are not effectively analyzed by two-dimensional gels because you can only load a small volume and you will only see the most abundant proteins. However, you'll note that these charge clusters and other trains are different molecular forms of the same protein. This streak here, for example, is our immunoglobulins. These are the various heavy chains and the immunoglobulin light chains. If I can get the precursor there, the immunoglobulin light chains are here. So you get some indication of heterogeneity of some of the abundant proteins, but you cannot even see medium abundance, let alone low abundance blood proteins. Protein interactions can be measured in several different ways. One is to isolate macromolecular complexes and identify the partners using LC-MS. Typically, this is done using affinity pull down, either using affinity tags or antibodies to one of the components, and then identify the components either in the entire protein, in the entire pull down, or specific bands from one-dimensional gels by comparing experimental control. There are a number of binary interaction analyses. One is protein arrays. Another is two-hybrid approach. Some newer techniques include biosensors in an array format where one can interrogate large numbers of proteins. So in summary, the most common types of proteome studies, the most common type of proteome study is protein profiling, which is quantitative comparison of two or more proteomes. The most time-effective protein profiling method is probably gel LC-MS for most samples. And gel LC-MS using current mass spectrometers can detect most gene products expressed in a single mammalian cell line. But it won't detect most protein isoforms resulting from alternative splices or alternative splicing or post-translational modifications. SILAC is the most precise and reproducible quantitative method, but it's limited to systems where metabolic labeling to steady state are feasible, although one can use a representative standard, such as a mixture of cell lines, to analyze a human tumor. The key thing is the representative standard must contain the vast majority of the proteins expected to be found in the specimen to be quantitated. Improvements to existing computational tools are important as these databases, as the experimental data sets continue to increase in size. So our last question of this lecture is, for which of the following proteome analysis problems would two-dimensional gels be a good choice? We mentioned that 2D gels are generally not preferred. So compositional analysis of human cancer cell line. This would be a particularly bad choice because, as mentioned previously, we do not get an ID of all the spots as part of the analysis. This will be a separate analysis. If I have thousands of spots, I have thousands of LC-MS IDs to perform. Analysis of a low abundance protein biomarker in cerebral spinal fluid. Cerebral spinal fluid is another biological fluid. It has some of the same problems that plasma does. That is, a few very high abundance proteins will prevent you from detecting low abundance proteins. Characterization of changes in phosphorylation levels, so an abundant 25K protein in purified mitochondria. By purifying mitochondria, my total proteome is substantially simplified over a cell line. Two-dimensional gel will be able to detect many of the proteins there. 25K protein that's very abundant should be easy to see, and because phosphorylation will change charge, I should see a charge train or cluster of charges at the same molecular weight. So this would be the best choice.